Before I start my talk, I would like to make a deal with you out in the audience. So your part of the deal would be that you should try to keep awake and you should have minimal interaction with your mobile phones and email. And if you keep your deal, my part of the deal would be at the end of the talk, try to describe to you why patients with chronic kidney disease are a little bit similar to these poor patients with Hutchinson-Gilford progeria syndrome. So almost everything you need to know about causes of aging you will find on these slides. You will have the causes of damage, genomic instability, telomere attrition, epigenetic alterations, and loss of proteostasis. Then you have the responses to damage. These are deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence. And finally, what is ultimately responsible for the functional decline with aging, these are stem cell exhaustion and altered intracellular communication. So senescence, or biological aging, is the gradual deterioration of functional characteristics in living organisms. And it's a normal part of an organism biology. The causes of aging are present throughout the entire lifespan, and unfortunately, there is no going back. It increases the risk for pathologies, morbidities, and mortality. And if you, for some magical reason, could stop senescence, the majority of us, maybe 95%, would reach 100 years of age. So if you don't die of senescence, there would be accidents, it would be suicides, famine, infections, and of course, wars. I think this is a devastating illustration of what war can do to a human being. This is a young Soviet soldier when he's entering the war in 1941, and after just 4 years of war, this is how he looks like. So really, war is not good for your health. And we all know that there is an increased risk of all this burden of lifestyle diseases with senescence. No surprise that you increase the risk of diabetes, of cancer, heart disease. But not all diseases accumulate with age. Asthma is an excellent example of a disease that actually decreases in prevalence with age. So we have a number of burden of lifestyle diseases that are depicted here. Chronic kidney disease is just one of them. They accumulate as we get older, and although they seem to be very, very different, they have two interesting things in common. The first part is a chronic smoldering inflammatory response that is thought to accelerate biological aging. This is commonly called inflammaging, and all these diseases that have an inflammaging response also associated with the loss of gut biodiversity. So in my talk, I will discuss quite a lot about the gut, and I will also discuss quite a lot about this cytoprotective transcription factor NRF2. And when we are stressed for various reasons, there is an upregulation of NRF2, and there is a number of anti-inflammatory and antioxidative genes that will start to work. I myself was convinced that NRF2 is of importance when I read this paper in JASN 2014. It's an integrative biology and transcriptional network paper in chronic kidney disease. And this study showed that all pathways aggregated into a network of two main clusters. That was inflammation and metabolism-related pathway. And NRF2 was served as the hub between these two clusters. So I believe if you are in a hub, it has some importance. And this transcription factor is constitutively expressed in all tissue, All the levels may vary among the organs. But when I did an extensive literature research of the diseases that accumulate with aging, irrespective if it's chronic kidney disease, if it is Alzheimer, cancer, diabetes, fatty liver disease, the NRF2 expression is downregulated. Its strong relation to the inflammaging response was presented in this Nature paper showing that NRF2 opposes transcriptional upregulation of pro-inflammatory cytokine genes. So there seems to be a specific NRF2-related diseasome with many different diseases, cardiovascular diseases, digestive system diseases, neurodegenerative diseases, respiratory diseases, as depicted here, that either they share genes, there are symptom similarities, and we all know that these diseases tend to co-aggregate. And this widespread nature of NRF2 may have an important therapeutic potential. So here you see in the center this two-component system with NRF2 and its inhibitor KEAP1. It's like a dimer switch that turn off and turn on hundreds of cytoprotective genes. And this will increase the clearance of oxidized and damaged proteins. And this transcription factor is something that you could affect by lifestyle. Psychosocial stress has been shown to downregulate NRF2. A Western diet, which I will talk quite a lot about, may also affect NRF2. Smoking downregulate NRF2. Exercise, which we know is good for you, has been shown repeatedly to upregulate NRF2. And then you have a number of synthetic NRF2 stimulators. Drugs that stimulate NRF2, the most well-known is bardoxolone, but you have also NRF2 stimulating activity in metformin, statin, and lithium. But what I find most interest is that they're in bioactive compounds, and especially fermented food. There are bioactive ingredients that have quite potent stimulatory effects on NRF2. The most potent of these bioactive nutrients is found in broccoli sprouts. It is called sulforaphane. And I was looking for a picture to illustrate the potency of sulforaphane, and I think this was the best picture I found. It has nothing to do with renal disease. But here you can see that in old rats, if you have dietary supplementation with sulforaphane, you can see the major impact on these rats. And this was via activation of the NRF2 KEAP pathway. So here you will have on the x-axis the chronological age, which we usually think about when we are having patients in front of us. On the y-axis is the biological age, which will be the most important age. So if we depict the ideal life course, that will be someone in which the biological age is all the time younger than the chronological age. These patients or persons, they will die at a high age without much disease. The average lifespan would be anyone that have a biological age that correspond to the chronological age. But the problematic group are those with premature aging. Patients with burden of lifestyle diseases for many, many years in which the biological age is considerably older than the chronological age. And this is the major problem in modern healthcare, this large gap between health span and lifespan. Because we know these patients with burden of lifestyle diseases, they have a lower quality of life, and there is a tremendous impact on the cost of healthcare. So there has been an intensive search to find viable biomarkers of biological age, especially vascular aging. There are a number of molecular and cellular biomarkers, DNA methylation, which I will mention quite soon. We have telomere length, we have somatic mutations, we have transcriptomic-based biomarkers, proteomic-based biomarkers. There are also functional and structural biomarkers of vascular age, for example, intima thickness, you have arterial stiffness, endothelial dysfunction, and there are also of some composite biomarker predictors. So at Karolinska, we have been interested to find markers of biological age, and this is one sub-study in about 200 patients in CKD stage 5, and you will find their chronological age in blue and their biological age in red, as estimated by the content of AGEs in the skin. And here you'll find a marked discrepancy. And actually, some of our patients, they had a biological age approaching 200 years, something that we really did not want to tell them. We also used a formula based on DNA methylation, and here you can see that there is a really nice correlation between the chronological age and the biological age in healthy controls. The patients in CKD stage 3 and 4, you will find a higher biological age as expected. But when we study patients with end-stage kidney disease, you will find that they reach the roof, and it's clearly they're reaching an unacceptable high biological age that may not be of clinical relevance. So recently we have been using a more accurate prediction of biological age, and these are the DNA methylation clocks. And in this study we used 3 separate clocks: the Horvath clock, the Hannum clock, and the new PhenoAge clock. The Horvath and Hannum clocks, they are the first generation clocks, and the PhenoAge clock is a second generation clock. And this method is based on that during stresses and as we get older, there is a methylation, especially if you have risk factors, and if you have protective factors, a lower biological age, there will be unmethylated sites. And I think the results came out quite interesting. I first should tell that this is a collaborative study together with Glasgow University. And here you will find a composite average of the 3 methylation clocks with the controls, and on the y-axis you will find an age acceleration. So here we can show that patients at start of dialysis or at the time point of transplantation, they have a higher biological age, about 4 to 5 years. 1 year of dialysis does not affect age acceleration, but interesting, there is a trend that is, and it is a significant difference, that after renal transplantation, there seems to be less of an age acceleration in chronic kidney disease. So I think you all agree with me that patients with chronic kidney disease, they are undergoing premature aging. And together with Jerome Coman and Peter Kontanko and others, we have been discussing in depth the impact of inflammation on aging processes in kidney disease. I think this figure is best depicting the content of this paper, that inflammation is something that will affect anti-aging mechanisms, for example, fetwin. It will, via stress response, stimulate catabolic pathways. Epigenetic changes and sympathico-vagal balance plays a major role. But at the end, all this will end up in this prematurely aged phenotype that we are all so accustomed to: muscle wasting, cardiovascular hypertrophy, osteoporosis, and last but not least, vascular calcification. And I will, this picture here, just very briefly show you that the strong relation between the telomere dysfunction and inflammation that exists in all these burden of lifestyle diseases. If you are inflamed, the telomeres get shorter. The shorter the telomeres, the easier you get inflamed, and this will be a vicious circle. So during our life, we are exposed to all the stresses of life. There is lifestyle habits, drugs, smoking, poor nutrition, infections, etc. And the stress of life will increase the allostatic overload that will, in all these burden of lifestyle diseases, stimulate this vicious circle that is so typical for the disease of aging. 4 main components of this vicious circle is NRF2 depletion, mitochondrial dysfunction, oxidative stress, and loss of gut biodiversity. And I would also think that tissue hypoxia plays a very important role here. So if you want to enable better aging, it is the exposome here that you should target. But now we are facing difficult times, and we have all these environmental risk factors: global warming, greenhouse gas emission, pollution, that also stimulate this disease of aging. It's already been shown that, for example, heat stress induces inflammation, oxidative stress, and premature senescence. And as this changes in the environment comes on very, very quick now, and the evolution of adaptive systems to cope with the rapidity of the changing anthropogenic environmental is so slow. We are not really protected against all this. But last week, there was an extremely interesting paper that was published in an ecology journal showing that in animals, you now see changing animal morphologies as a response to climate warming. So this is a real problem. And from Adira's home state, British Columbia, we are all aware of these alarming high temperatures during the summers. And this Lancet paper really in depth discussed the impact of hot weather and heat extremes on health risks. And of course, this global warming, heat events, air pollution will, especially in the vulnerable populations with high age, low socioeconomic status, impact our health. In nephrology, we are all aware of these epidemics of heat stress nephropathy that is related to climate change, and this is especially in mid-America and India and other hot areas of the world. So we are exposed to processed food, drugs, climate change, water shortage, sedentary lifestyle, which will increase the risk and stimulate this disease of aging. And we are in desperate need to find solutions and how to break this vicious circle. And here I think Rudolf Virchow said something really, really interesting many years ago, that between animals and human medicine, there are no dividing lines, nor should there be. The object is different, but the experience obtained constitutes the basis of all medicine. And the enormous diversity of species is actually one of the most striking aspects of life on our planet. And I believe this provides a source of solutions that have been developed during millions of years of evolution by natural selection in animals that live in extreme environments. And we recently published this review in which we in depth went through these age-related diseases associated with inflammation, and which is such an extreme high cost in our society, and find that there are many animal species that already developed ingenious solutions to combat these diseases. And this was already known by Albert Einstein in the 1920s. He stated that look deep into nature and then you will understand everything better. And this is this new discipline of biomimicry. What can we learn from nature? Elephants, for example, have developed ingenious mechanisms to protect themselves against cancer disease. And these findings are already in the way to find clinical applications. I myself have been very interested in hibernating bears, as they are metabolic magicians of definite interest for the nephrologist. Anyone that can survive well without peeing for 6 months should evoke the interest of nephrologists. Giraffes have blood pressure exceedingly high, 240, about 130, but they never develop stroke or kidney disease. And it was recently shown this spring that they have a specific mutation, and if by CRISPR technology they could insert this mutation into mice, they are protected against hypertension. The Burmese python snakes, they eat seldom, but when they eat, there are massive meals. They eat a whole goat or a pig. Just 1 or 2 days after these massive meals, there is a growth of their organs. There is a 40% increase in the ventricular mass. And this paper showed that there is a specific fatty acid combination that increase the mass of the heart. And this could be, of course, a really interesting treatment strategy for congestive heart failure. The wild tree shrews, they live in the jungle of Malaysia, and they eat fermented floral nectar as their main ingredient. Fermented floral nectar have a high alcohol content, so these tree shrews, they are constantly intoxicated with alcohol, but they do not develop the complications of alcoholism that we see in the humans. So over the past 350 million years, there has been an extremely complex interplay that developed between renal and hepatic metabolic energy and water conservation systems. And estivation is an extremely interesting process by which snails, crocodiles, hedgehogs, tortoise, birds, and lungfish enter a state of dormancy during the hot periods of the year. And this is to protect them and conserve energy and protect their organs. And in nephrology and cardiology, we have been overwhelmed by extremely interesting results regarding SGLT2 inhibitors as organ protectors. In this paper, Jens Titze and his colleagues extremely elegantly discussed the organ protection of SGLT2 inhibitors and the role of metabolic energy and water conservation, showing that SGLT2 inhibitors induce exactly the same estivation-like metabolic patterns, and this could contribute to the improvements in cardiac and renal function that we now observe with this class of therapeutics. So actually, nature had already figured out the way for organ protection in estivating mammals, and this is a metabolic switch that we humans now have learned to master in the 21st century. And I would find it extremely interesting to investigate if SGLT2 inhibitors could be one way to protect humans during future heat waves. So this is a picture of me in a bear den and looking for bears. And you may wonder why study bears. And the reason is that in bears we study not one but several simultaneous conditions that accumulate as we get older. If you and I would go to bed for 6 months and stay there, we would have problems with osteoporosis. We would lose more than 90% of our muscle mass. We should not be able to stand up on our legs. We would likely have thromboembolic complications and bed wounds. But in hibernating bears, despite 6 months of bed rest, there are now numerous papers showing that they do not get osteoporosis. They conserve muscle strength and maintain fatigue resistance extremely well. And during hibernation, when the heart rate is as low as 4 to 5 beats per minute, there are no risk for thromboembolic complications. And there are no bears that develop bear wounds. And it's interesting that during hibernation, they kind of have a perfect iatrogenic storm. And when we measure different parameters in the bears, you can see that besides being sedentary, they are insulin resistant. Some of them even have type 2 diabetes. They have kidney failure and they have extreme dyslipidemia. But their coronary arteries are totally clean and there are no signs of atherosclerosis. And this has been an enigma which we have been interested in. Phosphoryl plays a central role in oxidized LDL-induced immune activation in atherosclerosis. So antibodies against phosphoryl may be an atheroprotective mechanism, and this have also anti-inflammatory properties. If you look, for example, at patients with chronic kidney disease, their antibodies against phosphoryl choline are extremely low. But here we find that anti-phosphoryl choline antibodies, they are strikingly high in hibernating brown bears, especially look at the IgA anti-PC levels on the upper right corner. So this could actually represent a natural immunization process that prevent atherosclerosis during these high-risk conditions. So if we could find out why they have so high titers of this antiphosphoryl choline, that would be interesting. Could it be dietary effects? We know that they feast on a lot of berries in the autumn and summer. Could it be gut microbiota? And we also know that in the late summer and in the autumn, bears are infested by parasites and nematodes. And here I like to mention this Lancet paper, which I find extremely interesting because here they study indigenous South American Tsimane population. And in this population, they have chronic inflammation due to parasitic worms. So it has been speculated that parasitic worms could help keep arteries young, and actually our data here in the bears support this. And of course, in the long term, we will hope that this anti-PC levels may be something that could lead to a possibility to do vaccination against atherosclerotic disease. So in the human world, the longest living human being is this French woman, Jeanne Calment. She became 122.5 years old. She was born 1875, and when she was 12 years old, she actually met Vincent van Gogh, who came into her father's store to buy paint. And when she died, 1997, she had herself been working with internet. That's just an amazing lifespan to meet Vincent van Gogh and work on internet. And her secrets for being so long-lived could be she was very active. She played tennis, swimming, she was roller skating, she took up fencing when she was 85 years old. Eating habits also spectacular, drinking port wine every evening, and she would pour olive oil on all her foods and also liked chocolate. And then finally she quit smoking when she was 117 years old. But this is nothing against what you will find in the animal kingdom. Here we have the Greenland shark, which has a very strange name in Latin, Somniosus microcephalus. But they have a documented age, more than 400 years, and they become sexually mature when they are 200 to 250 years old. We have the Icelandic clam that live in the bottom of the ocean, extremely boring life, I would guess, for more than 500 years. Just imagine that you have a species with a heart that beats for 500 years. And together with Paul Shields in Glasgow, who is a gerontologist, we made a literature search to find out about animals that live for a long time and have negligible senescence. What do they have in common? We have, for example, the naked mole rats with a documented age more than 30 years, which are absolutely resistant to cancer and can endure anoxia for 20 minutes. We have this ocean quahog that lives for more than 500 years. We have fishes like the rotheye rockfish. We have the Greenland shark. And when you look at all these species together and find out what do they have in common, of course, there is little or no predation. They usually live in the cold milieu, but their NRF2 expression is increased. They seem to be superior in protecting protein homeostasis, and they also have highly efficient mitochondrial function during the lifespan. I think this could probably give some clues what will be relevant for human aging. And I think it's amazing that there are no signs of cardiovascular aging in the naked mole rat. Here is the pulse wave velocity measured in young, middle-aged, and old naked mole rats. No increased arterial stiffness. And in this PNAS paper from 2015, they wanted to find out what are the main difference between the ordinary laboratory mice, which have a similar weight to the naked mole rat, but they live only for maybe 4 years, while the naked mole rats become more than 30 years. And the main difference was a markedly increased expression of NRF2 due to a very low expression of KEAP1, the inhibitor of NRF2. Then this was both relevant both for the protein and RNA levels. So it has been speculated that NRF2 could be a guardian of health span and a gatekeeper of species longevity. And actually, if you look in papers in biology and ecology, you will find that NRF2 is consistently something that is a survival mechanism in the animal kingdom, irrespective if you look at European eels or the Antarctic silverfish or bats during hibernation, or in elephant seals that dive down to 2,000 meters for periods up to 2 hours, and they have absolutely no circulation in the kidneys, but they never developed acute kidney injury. Maybe this activation of NRF2 could play a role here. And I talked about the bears that could protect their muscles during this sedentary period during hibernation. And of course, also in bears, you will find that there is an upregulation of NRF2, and they could maintain and protect themselves against oxidative stress. So in order to understand all these burden of lifestyle diseases that accumulate with age, that are associated with inflammation and downregulation of NRF2, I think we need to study history, and we really need to go back a long time ago. So the great oxygenation event set the stage for an evolutionary transition to aerobe-dominated biota, and this continues to this day. And when this photosynthetic cyanobacteria started to produce oxygen in great amounts, we were exposed to oxygen. And when you are exposed to oxygen, there will be oxidative stress, and this could have some good effects for immune defense, cellular structure synthesis, but also oxidative stress increased the risk for mutations and cell damage. And the evolutionary origin of NRF2 is strongly allied exactly to the timing of the global transitions from anaerobic to aerobic conditions, because we needed to protect ourselves against oxidative stress. And as I already alluded to, there is a survival advantage in animals that live in extreme environments with an upregulation of NRF2, irrespective if you're talking about bears or bats or seals or Burmese python. But we can also get clues from clinical models of accelerated aging and markedly downregulated NRF2. Here we have Hutchinson-Gilford progeria syndrome, and what happened when you send NRF2 knockout mice to space. Because if you want to study aging, space is extremely interesting. And during long duration space flights, chronic space radiation exposure results in changes in telomere length and persistent DNA damage. And this was extremely well studied in these two twins, Scott and Mark Kelly. One of them stayed on Earth and the other one went into space. And then they could compare what happened to the transcriptome during this period. And in this Japanese study, they took NRF2 knockout mice and sent them into space for 31 days. And they find something really interesting because the space stress, because of cosmic radiation, microgravity, seems to be via the NRF2 deficiency. So space stress and NRF2 deficiency induces these aging-like changes of plasma metabolites. And they speculate that NRF2 plays an important role in regulation of adaptive responses to the stresses of space travel. So now I want to go more into the gut. And the reason of this is this cell metabolism paper, which I found extremely interesting, showing that gut-resident lactobacilli is a stimulus, something that can activate hepatic NRF2 activity. And they show that this gut-resident lactobacilli induce hepatic NRF2 in both Drosophila and mice. And this lactobacilli-derived metabolite with the very long name here activates NRF2. So there is a specific link between our microbiota and NRF2. And we know now that there are intimate connections between microbiota and organ functions, irrespective if it's immune defense, if it's metabolic factors, or if it's regarding the nervous system. And here we have a main problem in the industrialized Western world, because we are now adapting to a microbiota that has never before during evolution been experienced by Homo sapiens. And we will think that this is mostly due to what we eat. And of course, this may be the major factor, but studies like this one show that air pollution also affects gut microbial composition. Climate warming reduces gut microbial diversity. So this is a much bigger problem than just food. And during hunting and gathering in the earliest periods of life on planet, I think there was a very specific microbiota, and we can see the same time of microbiota in some remaining indigenous parts, tribes in Africa and Amazonas part. The naked mole rat has a gut microbiota that is extremely similar to what you find in these indigenous populations. There was a change in the microbiota probably during the ancient agricultural periods, and now with its modern Western diet, with processed food and a lot of drugs, there is a loss of biodiversity and function. And there may be something that we're now missing in our modern Western diet that actually could be protective, and this is discussed in this paper, that ancient foods that could actually have the disease preventive effects via something in the alkyl catechol that stimulate the NRF2 system. And during the human history, we have had a habit to fermented food to prolong the time when you can have to eat the food. And with fermentation, you actually invite the microbes you want and don't let in the ones you do not want. And I think this paper was just an eye-opener for me. It was published a couple of weeks ago in Cell by the group of Sonnenberg. And this is a prospective randomized multi-omics study in humans. And here they investigated the impact of fibers and fermented food. And they could show very elegantly that fermented food diet increases microbiome diversity and decrease the markers of inflammation. So we should eat more sauerkraut, more natto, and more kimchi. And this may be a way to reintroduce evolutionary important foodborne microbes that have been lost over the course of sanitizing the industrialized environment. Yeah, we live in a hungry planet and we need to eat. And here you will see pictures of what an ordinary family in different parts of the world eat during 1 week. And I think it's just fascinating to see how extremely different the diet is if you, for example, compare US to Ecuador in the right lower corner. And it's been clear now during the last 2 years that processed food, which is the key hallmark of Western diet, is not good for you. This paper showed that processed foods drives intestinal barrier permeability and microvascular disease. Here is a paper showing that ultra-processed diets causes you to eat more calories, and this will increase your weight. This paper showed that artificial sweeteners that you will probably take if you are diabetic, it induces glucose intolerance and it alters the gut microbiota. And when we have patients with chronic kidney disease, you will lose microbial biodiversity. These are yet unpublished data in collaboration with Glasgow, in which we analyze microbial DNA in patients with eGFR between 15 and 60, and then at end-stage kidney disease, eGFR below 50 mils per minute. And you can see the enormous loss of biodiversity in the gut when the chronic kidney disease progresses. And we already know from studies like this one in hemodialysis patients where you have a low biodiversity in the gut, there is an extremely high risk for death. Together with Port Shields, we also published these data. These are not patients with chronic kidney disease, but we're comparing populations in a high socioeconomic position compared with low socioeconomic position, and it was extremely clear that if you live in a high socioeconomic position area, you will have more circulatory salutogenic bacteria. And here you can find all these bacteria, and I find it interesting here that you will find that lactobacilla, which is a stimulator of NRF2, is actually 4-fold higher in the gut of the population that live in areas with a high socioeconomic position. And you could, based on this paper, read in Times, June 22nd this year, that unfriendly gut bacteria is linked to early aging. And this link is of immense interest for us nephrologists because we here had a number of microbial metabolites that is of much interest, especially 2 of them, trimethylamine N-oxide and phenylacetylglutamine, PAG, have attracted our interest. And we all know that there are many different mechanisms by which the gut microbiota may promote vascular disease, including short-chain fatty acids, lipopolysaccharides, and bile acids. But at this time, I will think that elevated levels and retention of TMAO and PAG in the uremic milieu and its effects on atherosclerosis and thromboembolic events is of major interest. And of course, these metabolites are related to residual renal function, but also to eating habits. So the gut microbiota plays an obligatory role in the formation of TMAO. It has been discussed, like here in JAMA 2 years ago, that TMAO is the new red meat risk factor. TMAO is produced from food rich in choline and carnitine. It is atherogenic, prothrombotic, and pro-inflammatory. The problem with red meat is that it is particularly high in L-carnitine. Red meat shifts the gut microbiome to produce much more of the precursor of TMAO, which is TMA, And on top of this, red meat also seems to reduce the ability of kidneys to excrete TMAO. TMAO passes the blood-brain barrier, and this paper showed that it promotes brain aging and cognitive impairment. This paper in the American Journal of Clinical Nutrition evaluated the impact of meat consumption on risk for dementia, and you can see that there is a weak effect here. But if you divide meat into processed and unprocessed meat, you will find that it is the processed meat that promote increased risk of incident dementia. So the processing of food may be detrimental, and we have all these numbers that the industry wants to put in our food for flavor and water holding, etc. But some of them have, of course, phosphate. We have sodium, we have potassium, but phosphate additives in food is a well-documented health risk factor. And here we make a very simple correlation between phosphate levels in the animal kingdom and their longevity, and you can see that there is a strong inverse correlation. And these are data based on the studies done by Makoto Kuro'u. And we all know that there is a relation, direct relation between the intake of phosphate and death. So here and now I would like to shortly discuss people living in Glasgow and the big cats, tigers and felids. What do they have in common? And here again we come to Glasgow with these two suburbs, one with a very high socioeconomic condition in which the average male will become 82 years old. 14 kilometers from Lenzie you will go to Carlton, and here the average male will only become 54 years old. So there's a difference in age in 28 years, only 14 kilometers apart. A lot of epidemiological studies have been shown that this population in Carlton, they have an imbalanced diet, they eat a lot of processed red meat, they have hyperphosphatemia, they have a high risk for cancer, and they have, of course, microbial dysbiosis. The press read this and stated that cheap red meat helps to kill off Glasgow's poorest men 30 years early. And here we have the big cats that also are extremely susceptible to chronic kidney disease. If you look at tigers and lions in zoos, 80 to 90% of them may have signs of renal dysfunction. Zoologists tell me that the most common cause of death in these animals are chronic kidney disease. And again, if we take the tigers and put it on this slope here with phosphate levels, you will find a perfect match. Just a couple of words about early vascular aging, which is something that we are working on in our laboratory at the Karolinska Institute using arterial biopsies from patients undergoing living donor transplantation, score them according to calcification, and we find, of course, that they have a low expression of NRF2, and this aligns with the literature showing if you stimulate NRF2 with various treatment strategies, this will actually improve vascular calcification. Another feature in these arteries is high expression of P16, P21 as marker of senescence, and this links us then to this extremely interesting area of senescence and health span. So cells become senescent as we become older, but if we are exposed to a lot of stress and diseases, more and more cells will become senescent, and the major problem with senescent cells is that they spit out pro-inflammatory signals that poison the surrounding tissue and promote disease. So a way to target senescent cells and make them senescent cells go into apoptosis is an emerging target for all diseases of aging, and that this concept actually works has been shown in several papers such as this one in Nature Medicine, showing that administration of senolytics actually have effects on both senescent cell transplanted young mice and naturally aged mice, and you can actually alleviate physical dysfunction and increase post-treatment survival by 36%. So as I promised in our deal from the beginning, I will now like to describe to you what patients with chronic kidney disease have in common with this extremely rare disease of Hutchinson-Gilford-Progeria syndrome. You will find not many of these children on the planet. It's only 1 in 4 million that have this mutation. And this causes an aging-like manifestations in childhood, and these children, they die of cardiovascular disease when they are teenagers. And this is a mutation in the LMNA gene that encodes for lamin, and there will be an accumulation of mutated lamin that is called progrin, and it is a mutation especially in 1824 mutation. And interestingly, if you compare these children with our patients with chronic kidney disease, you will find an extremely similar vascular phenotype and vascular pathology. It's been shown that progrin accelerates atherosclerosis by inducing endoplasmic reticulum stress in the vascular smooth muscle cells. And this Cell paper actually showed that suppressed activity of NRF2 and oxidative stress could completely recapitulate the Hutchinson-Gilford-Progeria syndrome aging defects. And if you reactivate NRF2, this decreases oxidative stress and reverses the cellular defects in Hutchinson-Gilford-Progeria syndrome. So the theory is that this mutated lamin A progrin actually entraps NRF2 and local restriction, and this causes impaired oxidative stress response and genomic instability. So in a study that has been ongoing now for many years, we are now comparing the vessels of patients with chronic kidney disease with controls, and we can see that also these patients, they express progrin. They have an extensive DNA damage, and interestingly, the exactly same mutation as you will find in Hutchinson-Gilford-Progeria syndrome was detected in the blood of all chronic kidney disease patients, and it was much more common than in the controls. So somatic mutations may actually become a risk when increased tissue regeneration is induced, and it provides some novel mechanistic insights into the phenotype of the premature vascular aging syndrome. So why are somatic mutations much more common in the uremic milieu? We don't know. But we would guess that uremic milieu, with all the retention of uremic solutes, will stress DNA and also may increase replication in certain hotspots. So, trying to end now, what did I tell you? I told you that loss of renal function leads to a mismatch between chronological and biological age. Rapidly emerging environmental threats, such as global warming, air pollution, stimulate the disease of aging. Animals that live in extreme environments, they have high expression of NRF2. That could be a survival advantage. I told you that unfriendly gut bacteria is linked to early aging, and the skewing in the Western diet towards more red meat and more processed diet has resulted in a dietary association with the disease of aging. And the role of somatic mutations, I believe, need much more attention in chronic kidney disease. And I'd like to thank the group involved in early vascular aging, here especially Carolina Kublicini and Anna Vitas in the lab. We have the group that work with food as medicine, Denis Mafra. Carla Avesani and Marie Evans will now start a study testing the effects of broccoli sprouts in patients with chronic kidney disease, and it's a randomized controlled trial. And then we have the biometric group here, and here we are, of course, collaborating closely with zoologists and veterinarians to get access to all these interesting animals. And Mary Lasker, she stated that if you think research is expensive, you should try disease. And therefore, I'm really grateful for the research grants to cover these problems.